Okay, it's gone 10 o'clock, so I think we'll make a start now. Okay, so I'd like to welcome everybody to our contribution to the Invasive Species Week webinars. We're going to be presenting a webinar on what lies beneath practical application in marine invasive and non-native species management. Just as a way of introduction, my name is Paul Stebbing. I joined Apem in 2019 as the invasive species lead. I have previous roles including providing advice to DEFRA on the implementation of the Marine Strategy Framework Directive. I also led the OSPAR expert working group on non-native species, as well as the UK Marine Group as well. So just to give you a running order for today, I'll be kicking off with an overview of invasive species in the marine environment, followed by my colleague, Dr Chris Ashelby, who will be talking about monitoring for marine invasive and non-native species, followed by Marc Hubble, who will be talking about risk assessments and biosecurity planning for marine invasive and non-native species. And then we have our guest talker, Jenni Kakkonen from the Harbour Authority, the Orkney Harbour Council, who will be talking about the ballast water management policy that they have in place. This will be followed by a dedicated Q&A session at the end. You can either submit questions via the Q&A menu item throughout the webinar, or leave it until the end and raise a hand using the reactions button at the bottom. As you may have noticed already, we have your microphones set on mute throughout, and then we'll, as you raise your hand, we'll unmute you so you'll be able to present questions at the end. So a recording of today's session is being made, and we'll be sharing it via email on Wednesday the 22nd. We'll be off camera until we finish presenting, and then during the Q&A sessions we'll turn our cameras on and go face to face. Just a brief introduction to who we are as Apem. So Apem is Europe's leading independent environmental consultancy specialising in freshwater and marine ecology and aerial surveys. We use expert evidence-based advice to meet clients' needs, working across a broad range of sectors. We have a dedicated invasive and non-native species team, which I lead, working across the marine, freshwater and terrestrial ecosystems. So if I move on to the introduction to marine invasive species. So just a brief covering of terminology. So non-native species are organisms which have been introduced outside of their native range by human activities that may survive and then subsequently reproduce. While invasive species are those non-native species which spread, causing environmental, social and economic damage. This includes both whole organisms as well as gametes, seeds, eggs, and also vegetative fragments. Within this, we haven't included diseases, although some people argue that diseases should also be included in this definition as well. There's one other definition, which is cryptogenic species, which are those that are of uncertain origin, so you don't know if they're native or non-native. A few examples of invasive and non-native species found in the UK are the crab, Hemigrapsis taconoi, the Cropidula faunacata, and the sea squirt, Didenum vexillum. So why non-native species are a particular concern? Well, people have been moving around the globe for millennia, and with that they transport species either deliberately or accidentally. And this has always resulted in the introduction of non-native species. As people have become more effective and efficient at moving around the globe, so for example, with transatlantic flights becoming a thing during the late 1950s, we've become better at moving invasive species, non-native species around, and this is seen, for example, with the Industrial Revolution around the 1800s, resulting in a sudden increase in globalization. And this shift in global economic force and global change promotes and diversifies trade routes, which further promotes the movement of invasive non-native species around. Now, a small proportion of non-native species may become invasive species and have an impact. So of the 2,000 non-native species currently found in the UK, about 2,000 species in the UK, about 200 of those are invasive. So that's about 10 to 15% of non-native species tend to be invasive. However, their impact is now so significant that they're considered to be one of the greatest threats to biodiversity worldwide, even more than pollution and climate change in some cases. So the impacts that invasive species can cause can vary considerably. So they can be ecosystem engineers. So, for example, Crassostria gigas or Maganella gigas in new money causes reefs and can change how habitats look and function. But also there's cases of indirect and direct competition with native species. So, for example, the Chinese mitten crab, Erythrea sinensis, competes directly with native species. They can also introduce diseases. So, for example, the American lobster introduced Gafkema into the country, which is a bacterial disease that affects native lobsters. The impact of any species can vary greatly across ecosystems and environmental gradients within ecosystems. So it's sometimes very difficult to predict or determine which species will have an impact and how they will have an impact. There's a clear challenge of understanding how ecosystems function is altered by the combined effect of species gained through invasion and losses through extinction caused by those introductions. But both of these are consequences and drivers of global change in biodiversity. Invasive species can also have economic impact. So, for example, they can impact ecosystem services, fishing, aquaculture, transport, and recreation. They can also have an impact on human health. And one of the main costs is in relation to remediation, as far as trying to reverse the impact that these species have. Just to present a summary of how invasions occur. So this is a simple conceptual model invasion process. So species will be picked up at a point of origin. They'll then be transported via a pathway to a new environment where they are introduced. And then sometimes, as discussed, these species can establish and then on rare occasions expand and an impact on the environment. Species traits and the physicochemical environment throughout this process may affect actually how these species, how invasive these species may be. So for example, species trait may dictate which pathways they move along and how they interact with any sort of community that they're introduced into, and then subsequently how they expand and where they expand to, while the physical chemical environment, so for example temperature or salinity, may affect the ability to move along a particular pathway and also expansion as well. Interestingly enough, actually the majority of invasive species or non-native species introduced are actually from areas that they've already invaded. This is called the bridgehead effect. Recent study conducted by the GB Non-Native Species Secretariat actually identified 8 top pathways, and of these relevant to the marine environment were hull fouling, ballast water, and movements of contaminated aquaculture stock. Invaders may actually remain innocuous for many years or even decades prior to having an actual impact. And this time lag inherent in many of the biological invasions imply that actually there are additional non-native species which have arrived within the UK, which may actually still cause problems within the future. This interaction between propagal pressure, the traits of invaders, the composition of recipient communities, and the physicochemical environment is largely unexplored and is really quite a complicated process. So as far as actually how management can be applied, this is the same conceptual model just turned on the side. Different management measures can be applied at different stages. So at the point of origin and the actual pathway, prevention of the species being picked up within that pathway is preferred course of action. But if a species is introduced, then rapid response to that introduction, trying to remove that initial pioneering population is essential. But as the population may become established, then trying to contain it, and then ultimately if the population expands, then actually trying to remove that population in its entirety. Management of marine species is considered very complex and difficult to achieve, especially normally there's an increase in cost as you go down this management ladder, as it were. And because of this but unfortunately, with the increase in cost, there's also a decrease in likelihood of success. And so therefore, there's a focus on pathway management, this old adage of an ounce of prevention is worth a pound of cure. And this is really illustrated within the legislative drivers. So, for example, the Marine Strategy Framework Directive and the Invasive Alien Species Order, focusing primarily on prevention. Historically, there has always been a focus on specific species, but there is now a shift towards this more pathway-focused management. And really, this pathway-focused management needs to identify and focus on these bridgehead regions, as mentioned before, as they tend to be the sources of many invasive species. But really, to be able to apply any sort of management, there's a need to actually understand and know what species are present, and be able to detect them early, but also understand and prioritise the most appropriate courses of action. And these are topics that my colleagues are going to talk about now. So I'll be passing over to Chris now.
Okay, thank you, Paul. So yeah, just to introduce myself, my name's Chris Ashelby. I work in Apem's marine laboratory, and I've been working doing marine surveys and marine sample analysis for about the past 20 odd years now. And my interest in non-native species really started when I found in my early days as a sort of junior scientist, found the prawn Palamon macrodactylus in one of my samples. So that sort of set me on the path of non-native species, but also crustaceans and prawns, which is what I did my PhD on. Okay, so monitoring for non-native species, as with all monitoring, really we need to ask a few questions, and the answers to those questions will help us decide the most appropriate and effective monitoring techniques to achieve the aims of our study. So, firstly, what is it that we want to monitor? Do we want to monitor a pathway, which is things like ballast water, hull fouling, shellfish movements, escape from aquaculture? As Paul mentioned, if we're monitoring a pathway and trying to prevent introductions at this stage, then that's the most cost-effective and most likelihood of success. Arguably, it provides the only opportunity to really detect and prevent the release of any new species. Or do we want to monitor a recipient environment? And if we do want to monitor a recipient environment, then we need to find out, what does the environment look like? Is it a natural environment, or is it a modified environment? Is it artificial habitat such as a dock? If it's modified, then what sort of modifications have we got? Have we got hard substrates now, like walls and marker buoys and jetties that weren't there previously? And if so, how are they distributed? Are we interested in monitoring all of those habitats in our area, or are we just interested in focusing on certain habitats? Do we have both intertidal and subtidal? And if we're monitoring an environment, then at this stage, the species is already out, and it's very difficult at this point to eradicate or control it. And as Paul also mentioned, by the time they're detected, normally the species are actually established at this point. And then, what questions do we want to answer from our study? Do we want to find out, perhaps an entire list of non-native species that are present in our area, or are we looking specifically for any new arrivals? Maybe we already have our baseline, so new arrivals is what we're interested in. Maybe we've done a biosecurity risk assessment and now know that we've got the species of concern that we're interested in detecting. Maybe those species of concern have come out of a horizon scanning exercise. Or maybe, if we're looking for a specific set of species, are we trying to work out how they're distributed? If so, what scales are we looking at? Are we looking locally, regionally, nationally? And maybe we want to determine how effective any mitigation measures we put into place have been. Whether any eradication or containment attempts have been successful. And next, we're looking at what timescales are we working to. Are we taking a snapshot of what's there at the moment, or are we trying to look at changes over time? We know that early detection is normally critical to enable rapid response to newly arriving non-natives. So regular monitoring is a good idea, but how regular is regular? That could be context specific. For example, monitoring may need to be more frequent where we've got an area with a large number of potential vectors than it would do in an area with very few pressures. And also in terms of timescales, we need to think about seasonal constraints. Planktonic communities, for example, change throughout the year, so you probably need to do monitoring on a regular basis in that case throughout the year. Algal communities die back in the winter, so if we were monitoring in the winter, then we find those were underrepresented in our samples. But as with most things, there's often a balance to be struck between our sampling strategy and data collection and available budgets. So we're going to look at some of the more common monitoring detection methods, and these are mostly applicable to sampling in the environment rather than a pathway. And it's probably worth also mentioning here that most of the sampling techniques are equally applicable to identifying native species as well as non-native. So if we're looking to maximize value for money, then really we should be trying to identify everything we encounter, not just the non-natives. And that, that can be particularly relevant, for example, if the species is not currently being recognized as non-native. So these might be the cryptogenic species that Paul mentioned. But in future it becomes recognized as non-natives, then we've already gathered records of that species. So rapid assessment surveys, probably the most commonly recognized method of monitoring for non-native species. By definition, they're a quick approach to monitoring. They're easily replicated between areas. So if we were looking to determine distribution on a national scale, for example, we can take a rapid assessment survey to different places around the coast and conduct a comparable survey. Typically, it's a survey comprising a timed search of a confined area, and it normally focuses on a target list of species and concentrates on fouling communities. So these are sea squirts, bryozoans, macroalgae, etc. We wouldn't normally be collecting physical samples unless they're required for confirmation of identification, and these sort of surveys are normally focusing on the species that are large and distinctive enough to be recognized in the field. And you'd end up basically with a presence absence list of species for a site rather than quantitative data, although you may end up recording semi-quantitatively on a SAC4 scale. They also would have a tendency to overlook mobile and small species. So the next thing is settlement panels. So on the right here we have a settlement panel, obviously one that's not been used yet. They're essentially used to monitor the same fouling communities as the rapid assessments, but the crucial difference is that they would provide you with physical samples that can be used to detect the smaller species and provide quantitative data. Ordinarily, you'd use a number of panels distributed throughout the study area, and they're deployed for a set amount of time. You'd ideally deploy them in time of high recruitment, so late spring is normally a good time, and then you'd leave them for a set period of time, probably a minimum of 12 weeks, but the longer you leave them, really the better. The 3D structure of the panel attracts different species. So for example, you may get shade tolerant species on the bottom or the inside of the triangle, whereas you get the more light tolerant species on the side. One potential issue you can have with settlement panels is that if you've left it for 12 weeks, then any of the fouling species that occur on that panel at the end would be a maximum of 12 weeks old. So you can have issues with identification of underdeveloped or immature specimens. So that might be an occasion where you consider molecular confirmation, for example, of your identification. Scrape sampling, I mean, again, that targets fouling communities. There's no real reason that you wouldn't combine this with a rapid assessment survey, so that could be used to then ground truth the results of your field data. As the name suggests, it involves scraping fouling organisms from a substrate, so it could be a pontoon, a harbour wall, a navigation buoy, the hull of a boat. And ordinarily you take a scrape of a known dimension, so determining abundance per metre squared, for example, is an easy calculation from there. So moving on to slightly more modern technique, I guess, although it's been around for a long time, molecular methods, environmental DNA, so-called eDNA, is becoming increasingly common in the use of detection of non-native species. eDNA is basically looking for traces of DNA that are shed by organisms into the water column or the sediment that they live in. And it can, eDNA sampling can be easily incorporated into other monitoring programs for sort of relatively little expense. It can be quite a valuable tool in detecting presence of non-native species through targeted sampling. If we wanted to look for a particular species of concern, for example, and it can provide records of species at low population densities where physical specimens might not yet have been found. So then you could target monitoring effort to specific locations, for example. As well as eDNA, we may also want to look at DNA sequencing to confirm morphological identification of species. As already mentioned, we might look at that for immature specimens recorded on settlement panels, for example. Okay. Photography and underwater imagery. Underwater imagery has some drawbacks in terms of you need really good quality photos. It's quite susceptible to turbidity, so it's not as useful in places like estuaries and ports, which are often hotspots for non-native species. But it can be used for larger, more obvious species, and it can be used to help determine distribution. It wouldn't be useful, for example, for morphologically similar species. So bryozoans, for example, quite a lot of bryozoans look very similar. So you'd probably, an underwater image probably wouldn't help you determine the actual species. And you could use it for an inspection camera, for example, to assess the extent of hull fouling on vessels. That would help you assess the risk that they pose. And then standard photography, for example, in intertidal areas, can be particularly useful, and it offers opportunity to integrate citizen science. An example of that is the oldest records currently from the UK of the crab Hemigrapsis takanoi were actually taken by an amateur naturalist and posted on a website, and they were there for 6-7 years before they were actually accurately identified. But it was a useful, provide useful early records of that species. And then, of course, we shouldn't be overlooking traditional sampling methods for providing records of non-natives. These generalist sampling methods, they typically provide records that may not be incorporated into our other sampling strategies. So unlike the rapid assessments and scrapes, etc., which concentrate on fouling communities, these are going to be looking more at mobile and informal species. In fact, they would tend to undersample fouling species. And yeah, I mean, things like trawling and netting, for example, might provide records of more mobile species. And if you're already conducting grab sampling in an area, then yeah, you probably already are collecting records of non-native species. So, glancing an expert eye over the data can extract the records of those non-native species, and then they can be used in your biosecurity assessments, for example. An example of this, some of the most recently recognised non-natives in Britain are the date mussel Archaeoatula senhausia and the window shell Theora lubrica. Both of those were recorded first from the UK in grab samples that were taken for other purposes. The date mussel was found in samples taken for the Water Framework Directive monitoring programme, and Theora lubrica was found in grab samples taken for an impact assessment. But if we go back to our original questions of what do we want to know, if we're looking to find the total number of species, then really what we need to do is use a variety of sampling techniques. Basically because every species has a slightly different ecology and habitat preference, then the more sampling techniques we use, the more species we will detect. These figures from two different studies we've been involved in. The first one had 33 non-native species. The second one had 75 non-native species. Those figures do include cryptogenic species as well. But as you can see, there's not any single sampling type that records all of those species, and several of the sampling types have a species that was only recorded by that sampling type. So it was only really by combining lots of different methods that the complete list could be obtained. I'd argue that the list is probably still not complete. There's sampling types we haven't used in these areas, and for example, the fyke nets and seine nets that didn't record any unique species, there were only 2 or 3 seine net samples included in the data analysis, so that it's potentially undersampling or underrepresented at the moment. Okay, so that concludes the bit on monitoring. It was a bit of a whistle-stop tour, but I'll now hand over to Marc, who's going to talk about biosecurity risk assessments and biosecurity planning.
Thanks for that, Chris. So I'm Marc Hubble. I'm an Associate Director at Apem and Head of Marine Consultancy. And I've been at Apem for over 15 years now. So over that time, I've been involved in a very wide diversity of marine projects, many of which have involved consideration of non-native species. And more recently, many are involving requirements for biosecurity plans. So what is a marine biosecurity plan? Well, effectively, it's a document that combines the risk assessment, so looking at the potential risk of introduction and spread of non-native species associated with a plan or project, and the proposed management measures that are in place to control that risk as far as possible. So there's a number of steps that we need to go through. We need to be collating information related to construction and operational phases, looking at the pathways by which non-native species could be introduced or spread based on what sort of works are being conducted, an assessment of the risk associated with that, and then where there is a risk, looking at what control measures can be put in place to reduce that risk as far as possible. And then also it's necessary to have contingency in place. So if there's a breach of those control measures, what do we do then? So as Paul was mentioning, there's obviously a wide range of potential effects of non-native species, not least in terms of marine biodiversity. And by having a robust biosecurity plan in place, it's very much a demonstration of best practice by a developer. There can be effects on performance and operations in certain sectors. For example, Japanese wireweed, Sargassum muticum, can colonize fish cages and oyster trestles. Colonial sea squirts, for example, the carpet sea squirt, could colonize infrastructure and basically affecting or inhibiting performance and operation. And there's a wide range of policy and legislation associated with non-native species. For example, the Marine Strategy Framework Directive, Descriptor 2 of good environmental status, is associated with the effects of non-native species on marine ecosystems. for the Water Framework Directive and following the Clearing Waters for All guidance, there's a section in there where it's necessary to consider potential effects of non-native species. Other relevant legislation includes the Wildlife and Countryside Act, the EU Alien Species Regulation, and the Ballast Water Management Convention, which Jenni will talk about a bit later as well. So when conducting an ecological impact assessment, without mitigation, you would often consider the potential effects of non-native species could be significant. So the indication that you've got a robust biosecurity plan in place and the actions within that plan are often considered sufficient to reduce that significance to an acceptable level. So for this reason, and the other reasons mentioned, they're often a requirement now of marine license applications as well. So the guidance that we tend to follow, written by Liz Cook and colleagues in 2015 for England and Wales, which is very much an edit of guidance written for Scotland in 2014, the Scottish Natural Heritage. And this outlines two types of plan. There's a site plan, which is more for long-term ongoing activities at a single site, and an operations plan, which is more for time-limited activities. Now, in reality, for a given project, you're generally considering a combination of these plans, whereby, slightly confusingly, the site plan aspects would be related to the operational phase, where you've got long-term activities, things like maintenance dredging, for example, and the operations plan is more associated with the construction phase aspects, which are more short-term. The main difference between the two is that for the site plan, you'd have measures and expectations in there for regular review and perhaps ongoing monitoring and the ability to modify that plan as you go along. So what do we need? Well, basically, in terms of information, we're looking at the kinds of methods that are involved for the work, the equipment that's going to be used. Is there interactions between equipment and the water column? What's the number of vessels, types of vessels, sizes, and their general movements and operations around the work site? For personnel, are there going to be interactions between personnel and the water column? When will the works be undertaken? How long will the works take to complete? The footprint of the works, and also the geographic location, so where is it all happening? Similarly, for operation, same sort of range of information, but more often that would likely be related to vessel movements at a site and maintenance activities. So we also need environmental information for the site. Are there any hard structures or man-made structures that would be particularly susceptible to colonization by non-native species? And also particularly interested in aspects such as water flow and salinity, which can affect the ability of non-native species to be established at a given site, depending on the species being considered. So we also need to get a good understanding of what non-native species are already present. So this is using the wide range of methods that Chris was mentioning, and also desk-based study, and ideally a combination of the two where possible. So once you've got all that information together, you're then looking to assign a risk level for the different activities involved in the works, and that will be assigned as high, medium, or low. And then for any measures, for any risks that are identified to be medium or higher, then it's necessary to look for appropriate and suitable control measures that can then be applied to reduce that risk as far as possible. And the contingency plan then needs to be drafted, which shows what to do if there's a breach of any of those measures, and also what to do if new non-native species are detected on site. So this is just an indication of how we present the information. The idea is very much for it to be simple, straightforward, very clear, very easy to follow. So for that reason, it's done in a tabulated structure. This is just taking one example for a project, which here it's the arrival of some of the construction vessels to site. Other aspects you'd consider would be related to ballast water exchange, introduction of structures to the water column, that kind of thing. So you've got the activity on the left there, then the risk that's involved, which is introduction of new non-native species. And then there's a number of factors to consider. So for this aspect, we're talking about biofouling of holes in general here, but obviously biofouling of niche areas is also a consideration. So in the Cook et al. Guidance, there's actually a very useful table which ranks different levels of biofouling from 1 to 5. For example, you could have no biofilm or very slight level of biofilm. And with that, you could consider the risk of non-native species being present and therefore potentially introduced or spread would be low. But if you've got a ranking level of 3, which equates to more than 5% of an area being biofouled with more than one type of animal present, then maybe the likelihood of a non-native species being present is higher, and therefore the risk of introduction and spread is higher. We also need to take account of the amount of time that a vessel is present at site. So the longer that a heavily biofouled vessel is present at location, then the more chance there is of non-native species, if they're present, of being introduced to the local environment. So you allocate those different levels of risk to the tasks, and then you put the overall or the worst case risk, if you like, as the overall risk for that activity. Another consideration is geographic movements. So if there are vessels coming from worldwide locations, it's necessary to consider that the risk of potential introduction of non-natives is greater if you've got a vessel coming, for example, one temperate region to another, or similar environments, as opposed to a tropical region to a temperate region. And that sort of consideration can also readily be built into these risk assessment tables. So once you've identified the risk, then it's necessary to look at some control measures. So here we've just taken the single example we looked at previously of vessels arriving to site during the construction phase. So aspects to consider is could be ensured that vessels remain at the site during construction, so they're not leaving and returning from different water bodies during the construction phase. We'd look to see that biosecurity assessments are undertaken for all vessels, asking for anti-fouling treatment records, and also looking to see if International Marine Organization guidelines are being followed in terms of management of biofouling levels. If the biofouling levels are high, so ranked at level 3 or higher as an example, then it could be insisted that the vessel can't enter the site until that biofouling is removed. That can be done by different methods. It could be that there's mechanical removal where it's ensured that all material is readily contained and disposed of appropriately. A very effective method can be air drying. So if there's dry dock available, for example, to take out the vessel for at least 48 to 72 hours. And also chemicals could potentially be used as well. But what is used and what is most appropriate is entirely project and situation dependent, and also consideration of relevant logistics as well. So once you've got those measures in place, there needs to be someone in charge who's overseeing all of this and seeing what's being conducted. So there should be a biosecurity manager that's allocated to the construction phase and the operational phase. That could be an ecological clerk of works or a site manager. And they should have a logbook where they're recording a wide range of information. So whether there's any surveillance or monitoring that's been undertaken looking for non-native species, any training of staff, where they've been trained to look for and identify conspicuous and readily observable species, and also to report them. Then noting any control measures that have been put in place to reduce the risks, any anti-fouling records that are available, and also if any new non-native species and especially invasive species are recorded, to make notes of that. Then there also needs to be a very clear action plan, whereby if there is any breach of control measures, who is in charge for each individual control measure and breach, and what would they need to do in that situation. And then if any invasive non-native species are recorded, they should be reported immediately to the GB Non-Native Species Secretariat. And then next steps considered. So is it looking at possible options for containment or eradication, whereby eradication is obviously very difficult. But there are options available for those, and the next steps would be discussed with relevant stakeholders as well. So that's basically a summary, a brief summary of what's involved, and very much the idea is to have a very clear, easily followed document that outlines the assessment of the risk, the steps to be taken to control it, and this document can be readily presented by developers and provides evidence that they are, adhering to the best measures possible to reduce the risk of introduction and spread of invasive species. So that covers that section, and I'll now hand you over to Jenny, who's going to be talking about ballast water management, specifically in relation to operations at Scapa Flow.
Thank you, Mark. Hello, everyone. I'm Jenni Kakkonen, and I work as a marine biologist with the Orkney Harbour Authority. And I've been working at the Harbour Authority for over 14 years, and I lead all of the marine environmental monitoring programs for the Harbour Authority, which includes non-native species monitoring and ballast water monitoring. And today I'll be presenting on our ballast water management policy and our ballast water sampling program. I'll start giving by a little bit of background to the ship-to-ship operations in Scapa Flow. So Scapa Flow is a large natural sheltered anchorage that has been used as an STS site since the 1980s. And the ballast water policy has been in place for Scapa Flow since the 1980s. About 10 years ago, the council realized that this ballast water policy was both out of date and commercially unviable. And in 2010, the process to up-to-date the ballast water policy was started. So here's some of the main steps that were taken to update the policy. The IMO Ballast Water Management Convention has its approved ballast water exchange areas. And very early on in the Orkney policy development, it was realized that we would need to establish an Orkney-specific exchange zone in the North Sea. To be able to do that, a full mathematical model of tides and currents in the North Sea and around Orkney was generated to understand the impact of exchanging ballast water in the area. This was also done to understand the impact of discharging ballast water into Scapa Flow, and a detailed model was also generated for Scapa Flow. After checking and revising the original strategic environmental assessment and environmental impact assessment in 2010, it became obvious that under the habitats regulation appraisal requirements, that an appropriate assessment of a revised ballast water policy would also be required. The check and integration of the IMO 2004 convention into an IMO EU compliant ballast water management policy was also done. During this up-to-date, there was numerous consultation events with local and national bodies, organisations, government advisory bodies, public seminars, and with the members of the Orkney Island Council. In 2013, non-native species baseline study was also carried out in Scapa Flow in order to determine the presence of any non-native species. And this was done using historical records and collection of samples at locations as per their policy. So after all of this, we had an integrated ballast water management policy, which took into account the IMO Ballast Water Management Convention and the relevant European legislation, and resulted in the Orkney Harbour Authority approved ballast water management policy. So the policy highlights are that it was approved by the Orkney Islands Council in December 2013 and is IMO compliant with regular reviews and updates. The approved ballast water management policy clearly states that all vessels will need to do exchange and treat if the vessel is fitted with approved treatment system, and there's always an exchange process as per approved policy. The policy includes a special exchange zone for vessels approaching from the east, and this is called the Eastern Exchange Zone, and vessels from the west are to be IMO Ballast Water Management Convention compliant. The full SEA and HRA was completed, and this concluded that beyond reasonable scientific doubt that there would not be any adverse effects on the integrity of European sites, and in this case, this site was Locos Tenus. The policy includes both a ballast water monitoring program as a way of ensuring compliance with the policy, and also a marine non-native species monitoring program to monitor the receiving environment. So just to recap the IMO regulations D1 and D2. So the D1 is the ballast water exchange standard, which is either 3-time volumetric exchange and normally is a flow through, or empty and refill. And the regulation D2 is ballast water performance standards, and this is the discharge concentration standard which all approved ballast water treatment systems is meant to discharge, and it has plankton classes in both small and large size fraction and indicator microbes. So in here you can see the OIC approved ballast water exchange area in the North and North Sea. It's the hashed area and is called the Eastern Exchange Zone. And you can also see the IMO approved areas which excludes all coastal zones. So just to recap, Orkney is in north of Scotland mainland, and Scapa Flow is the area in the southern Orkney Islands. The ballast water management policy states and has 4 ship-to-ship transfer locations, which are marked here in yellow, and we also have 11 anchorages. So basically, the enforcement of the policy includes work done before ballast water sampling happens, and on the ballast water and on the day for the ballast water sampling. So as soon as a ship-to-ship transfer is confirmed and we get an ETA for our vessel, the vessel movement is tracked by the Harbour Authority using AIS, Q88 and IHS. And the ballast water exchange location is always checked, and this is done using the AIS and as well as the IMO ballast water reporting form, which is submitted by the vessel before they arrive and after the ballast water exchange. On the day of the ballast water sampling, a Harbour Authority representative boards the vessel to check all documentation. At the same time, a ballast water sampling team boards the vessel. The first thing that the sampling team does is to collect salinity samples from 2 different ballast water tanks, and these are tested on board straight away by the Harbour Authority representative. If the salinity is within our guideline limits, the full sampling of ballast water continues, and only after the ballast water samples and the documents are checked, permission to discharge ballast water is given. So we have had 3 different types of vessels in Scapa Flow that have required ballast water testing. We have accommodation vessels, which are semi-submersible. We have also had a few drilling rigs, then liquefied gas carriers and crude oil tankers. Each one of these vessels require different type of sampling methods. Today I will be concentrating on the methods that we use for the crude oil tankers. So all of our sampling is carried out on deck, regardless if there is treatment system or not, and that's for operational reasons. We collect salinity and indicator microbes using a bailer from the middle water depth within the tank, and these samples are tested in-house at Orkney Harbour Authority. Plankton samples are traditionally just collected using plankton nets. Ideally, they would be lowered down into the tank and collected a full depth of the tank. But nowadays, because of health and safety reasons, most of the vessels have an interdeck, and because of that, we have started using hand pumps. And the reason it's a hand pump is there's no electrical equipment allowed on deck of an oil tanker. So basically the water is pumped from the tanks and concentrated using the plankton nets, and these are collected from the middle depth of the water tank, and we collect both small size fraction samples and large size fraction samples for both phytoplankton and zooplankton. And the phytoplankton samples are preserved in glucose, zooplankton samples are stained with neutral red and preserved in 4% formalin. All of the plankton samples are sent to a taxonomic laboratory, which is Apem, for analysis, and they are analysed for viable and non-viable organisms and identified to species level if possible. So here you can see the number of vessels that we have carried out or tried to carry out ballast water sampling since 2013 to 2019. We received the oil transfer license in 2015, and you can see how the numbers of the vessels coming to Scapa Flow have steadily increased since then. You can also notice that the vessels with the ballast water treatment systems that are operational have been increasing since 2018, and that's highlighted on the yellow bars. So this is some of the information that we collect from each one of the vessels that come to Scapa Flow. This is part of the reporting forms, and here you can see that the last port of call with most of the vessels that come to Scapa Flow is in southern North Sea, and the favourite ballast water exchange location is the eastern exchange zone in the northern North Sea. And this really highlights that when we were in the development phase of the policy, it was really important us to explore and set our own ballast water exchange area in the northern North Sea. And here's some of the results from the first 146 sampling events. So this is up to December 2019. And as you can see, most of the vessels salinity is within normal seawater range for Scapa Flow in North Sea, which is 35 PSU. And for the indicator microbes, we've only been looking at the E. coli and enterococci. E. coli, all of them, the samples have passed the D2 standard, whereas in enterococci, there's only been 6 samples out of 453 that has been tested that has failed. So we don't have any concerns regarding salinity and indicator microbes for the ballast water that comes to Scapa Flow. We've also looked at some of the trends within the plankton communities that is in the ballast water. We've decided to divide the phytoplankton and zooplankton communities for this analysis, and we looked at 3 different variables. So it's the last port of call, exchange zone, and time, the month of the ballast water was exchanged. And basically we wanted to see if there was any trends in the ballast water coming from looking at these three different variables. And as you can see here, for both phytoplankton and zooplankton communities, they are very clustered, so there's no trends as for the last port of call for the vessels. Very similar results for the exchange zone. So here we have Eastern Exchange Zone, west of Scotland and other, and again, there's no trends at all evident in these CCA plots. And again, same for the month of sampling. All the samples are very clustered, and there's no trends at all evident in these samples. So in conclusion, the meshing of the IMO and EU requirements into one policy was a very long and difficult process. And it took us several years to fully understand the nuances of what is involved in a ballast water sampling on board vessels in operational and very busy harbour. All the vessels that come to Scapa Flow have been very happy to comply with our ballast water management policy. And I really want to make a point that the ballast water sampling and the enforcement of the policy, it really is a team effort. There's the Marine Environmental Unit, the sampling team, the Harbour Authority representative, pilot boat and crew, taxonomy laboratory, plus the VTS ops team, and so it goes. So it really is a team effort to get all of this done. Both the development of the policy and the ongoing ballast water sampling program are very costly, and both are covered by the STS income. And just looking at the data, there really wasn't any trends in the plankton communities when compared to the last port of call, exchange area and month of sampling. And this indicates that the risk of possible introduction is the same and not dependent on any of the above factors. And we really wouldn't be accepting any exemptions for any of the routes at all. Thank you very much. I'll hand you back to Paul.
Brilliant, that's great. Thank you ever so much, Chris, Marc and Jenny. So that's the end of the presentations. Does anybody actually have any questions they'd like to present to the panelists? If you'd like to raise your hand, then what I'll do is invite you to unmute. Sarah, you can ask a question.
Hi there, thank you very much for those really great presentations. I'm in the Environment Agency working on applying the Clearing Waters guidance with various developers and industry. I'd wondered if you thought more could be done to improve the Clearing the Waters guidance for all to help reduce some of the impacts of invasive non-natives in the marine environment.
I mean, at the moment, it mainly indicates that they need to be considered, doesn't it? And it's not, and it's down to the very much the interpretation of those producing the Water Framework Directive assessment, how they address that and how they present that information. We've not had any comebacks from the EA on the way that we've presented it in Water Framework Directive assessments that we've produced to date, but we do very much point to the fact that there'll be a biosecurity plan in place outlining key measures to ensure that there are limited effects on the Water Framework Directive water bodies. And sometimes it's necessary to present that plan at the same time as the Water Framework Directive assessment, but sometimes it isn't, or it might be agreed that that's something that can be provided, down the line to the satisfaction of the EA. So it is quite open and flexible at the moment as to how it is to be able to address that side of things. But I don't know that that's problematic. I mean, what are your thoughts on that?
I would imagine, just seeing particularly the Orkney example there, I mean, we've got a very robust approach. My own impression is that we probably have very inconsistent ways of doing things around the country, and that's probably lack of experience in applying some of the guidance in different places for different activities. So I think there's a bit more that we as an organisation can do in the Environment Agency to be clearer on what we would expect to see.
Do you find you give much feedback on what you do receive? So if you do get something and you're not sure that it's quite adequate, would you go back and say that?
My team and my personal experiences, we have done, but I don't have. We haven't had those sort of discussions in the wider agency to know people's approach more generally. This is stimulating me to maybe start that discussion.
That's probably a good thing.
Thank you.
Okay.
Thanks, Sarah. Yeah, has anybody got any other questions they'd like to raise? I don't know if the panelists have any questions they'd like to ask.
Are there no other hands up?
Not that I can see, no.
Okay, I just had one for Jenny actually, because you're indicating there one of the things that you were looking at was recording the salinity in the ballast water tanks. And I just wanted to check with you the main reasons for that, or what you're looking for. So if the salinity is lower than you're anticipating, is that demonstrating to you maybe that they haven't taken the ballast water from where it was said it was going to be, or there's going to be increased risk then of introducing of non-native species? Is that what you're looking for?
Thanks, Mark. Yeah, so basically that's exactly, so we have a set guideline limit, so for the salinity that we're looking at, so it's 30.5 to 35.5, and it has to be within that range for us to accept it. And if the salinity is less than 30.5, we would consider that it could potentially be coastal water. So, and the IMO Ballast Water Management Convention specifically said that ballast water exchange shouldn't be done in the coastal waters. So what we would do is that we would reactively straight away sample more tanks on the vessels to see if it was just a blip or if it was consistently too low. And then we would potentially send the vessel back out to sea to do the ballast water exchange in the correct area to kind of avoid any invasive species entering. And we have done that in a couple of occasions, and the vessels soon get the message that they really should do that. But there are some occasions where it's been quite low or in the limits, and we have checked all the documents and the AIS track, and we have accepted that as a correct, and it could have just been a partially incomplete exchange. But yeah, so we definitely look into that, and the same goes into the higher ranges, because, for example, Mediterranean is very, very high salinity, so we would not want any of the non-native species or invasive species from the Mediterranean area to scupper flow. So it works both ways.
Okay.
Yeah, really interesting practical application of the of what's going on with the convention. But does anybody else have any questions? If they'd like to raise a hand now. Okay, I don't think there's any other questions to be had. And if there are any further questions, then please feel free to drop us an email if there's any that occur after the actual session. As I said previously, the recording will be released on the 2nd of June for everybody. So yeah, thank you ever so much for everybody attending. Really greatly appreciated, and I hope you enjoy the rest of Invasive Species Week. No.
Thank you.
Thank you very much.
Thanks.
Thank you.
Thank you.